Dear Stefan and Dean, I'm so glad to see you again. How are you doing?
Oh, every time better.
Me too, thank you.
I want to quickly introduce myself and my guests for those who don't know. My name is Kate Sugak. I'm a naturopath and an activist who is actively working on attracting public attention to the fact that virology is not based on scientific methods. And my guests today are Stefan Lanka, who is a microbiologist and virologist, but he calls himself a non-virologist, and you will understand why in a minute. And his close associate, Dean Browse, who is working together with Stefan to help humanity awaken to the truth about health and disease and to the real biology. So today, Stefan will announce us something very exciting, which is the results of the second phase of his control experiments for SARS-CoV-2, which are the final point in the refutal of the whole concept of virology. Stefan, to help people to understand better the importance of it, could you please explain what are the control experiments in science? For what purpose did you initiate these control experiments for SARS-CoV-2? What was achieved in the first and the second phase, and why it is the end of virology?
Yeah, thank you very much to have the opportunity to explain those most important things. I mean, science is to prove something. And when you are using techniques and not pure logic, that you do some experiments, you have to show and to document that the technique you are using, it's not causing the results or disturbing the results. So, and the rules of science have been written down in the year 1998. And they are part of the working contract of every scientist who receives money from the state, so tax money. And they are forced to publish to the full extent the control experiments. So, but in the whole field of virology, there are no documented control experiments. Sometimes they mention they did it, but it's just a word and no explanation what in fact they did, no documentation of the steps of the experiments, nothing. And especially nowadays, when a virus is said to be proven when they publish a genetic sequence, there are none and absolutely zero control experiments in this whole field. What they say is molecular genetics. And this is easy. You're just reading those papers and you'll find, no, the word control experiment doesn't appear. So, and in order to disprove virology, so I call myself now an ex-virologist, because the structure I isolated 30 years ago is a kind of mini spore, but not with a negative potential. It's just smaller than real spores, which can outgrow, grow to build a new organism. Those mini spores, they have a metabolism on their own, but at all, they are a piece of genetic material surrounded by a shell. And those mini spores do exist in a great number in the oceans, and they came to be known as the bacteriophages in bacteria. And bacteria have no chance to build spores because the situation is changing too quickly for them to build real spores. Then they build a kind of mini spores, and those mini spores called phages in bacteria. And those phages in bacteria became the model how viruses should look like. And here the whole thing started. What the people don't know, that we had an old virology where a virus was considered to be a piece of protein, a piece of toxic protein. And that this protein has the ability to self-replicate. So the genetic material was thought to be a protein itself. So proteins were said to be able to replicate on their own. But then doing studies in 52 with the bacteriophages, it became clear that every protein needs a piece of nucleic acid to be synthesized. So it needs a nucleic acid as a catalyst that a protein is coming into existence. And from this moment on, the people were thinking, oh, those nucleic acids are the genetic material, which they are forced to think that we have a genetic material. Because in our science, since 2,500 years, it's only allowed to explain life and disease and processes of healing in terms of pure materialistic interaction. And this model was even sharpened, got more crazier in the year 1858, when it was declared that life consists only out of cells, and that only by cell division an organism comes into existence. And from this time on, the people were forced to think that there must be a genetic material in every cell. So this where we had the notion, oh, there must be a genetic material. Till 52, it was the idea, proteins are the genetic material. Then in the study of bacteriophages, they realized, no, every protein needs a nucleic acid to be synthesized. And from this time on, there was the idea, oh, nucleic acids are the genetic material. And they were forced to think it's a book of life. When out of a cell, one time comes an elephant, the other time a tree, and out of the other one, eukaryote. So we are forced to think there must be a genetic material. But this was disproven from the year 2000 on, because it was become clear that the genetic material is ever changing. Every nucleus has a different kind of genetic material. So, but in the public, they were saying, no, we have one and it's stable. And what the geneticists are doing was copied by the virologist, because from 52 on, the idea was, oh, a virus is a piece of genetic material, a dangerous one. Naked, the pure ones in Africa, Ebola, or the ones with envelopes. And they started to do the experiments, and they thought when they kill tissues in the test tube and they transform into molecules, they thought, those molecules are viral. Even so, they never could find a complete virus. They were not able to photograph something. All photos shown to us are not out of a human being or an animal, out of their liquid, out of their tissues. They are only artifacts in test tubes. And here the control experiments start, which they never carried out. You have to treat cells in the test tube in the same manner like you would do it when you think, oh, it's infected. And then to see if you see a difference. And no, the same thing happened. And this was phase one of our control experiments, that we use healthy human tissue and treat it exactly the same way virologists were doing when they believe, now we are infecting the cells in the test tube. So we did exactly the same, and we get the same results like when virologists are convinced, now this is a virus. And this first phase of the control experiments was disproving the whole field of virology. Because since 54, this idea was established: when cells are dying in the test tube, it must be a virus. And this never ever was controlled. Why? Because this idea was established by somebody who half a year later, after he published this idea, and he published that is an idea full with contradictions, but then he became the Nobel Prize for another thing, another idea. And with this Nobel Prize, this speculation became a scientific fact and the basis of the whole new virology, after the old virology gave up themselves in the year 52. So we have the birth of the new virology, where virology was, a virus is defined as a genetic material of a given size, of a given sequence, naked or with an envelope around. So, and these control experiments we published in the issue number 2 this year of the magazine Wissenschaft plus Science Creates Plus. So, and the second phase of the experiment, we took the nucleic acid out of the healthy human tissue, we multiply them, and when we multiply them in a very strict scientific manner, that means only 12 times, we'll get 20% of the genome of SARS-CoV-2. And when we use 30 cycles, which is considered in biochemistry as already dirty and not scientific, we'll get, when we use the same error rate, like virologists are using, we'll get 100% of the genome. And virologists, can you imagine, they are using 40 to 45 cycles. And what they are using, they add to the cells a lot of synthesized small molecules with given sequence, such a high amount that they add already 20% of the sequences later. They are reading and say, look, this is part of the virus. And this is what the control experiment in the second phase were showing, that all this, what they are saying is the genetic material of a virus comes out of normal human tissue. It's nothing specific for something which comes from the outside. It's pure human sequences. And we use the same techniques virologists are using, and we'll get the same result. And that means this is the disproval of all this speaking and claiming that we have a viral genetic material and that we can detect it with PCR in a body. This has been disproven with the second phase of our experiments, and they are going to be published in 6 to 8 weeks. We have this result published. And above all, what is showing that virologists never ever came up when they are looking for a new virus, even if they multiply in a very dirty way, in the first round of multiplication, they even then are not able to construct with their software tools the complete genome. So they arrange what they multiplied in a very dirty way, and then they synthesize the primers and do a second round of a very dirty multiplication. And this you must imagine. Without multiplication, they find nothing. With the first round of multiplication, they find no complete genome. And they do a second round, the same time way dirty, and they add synthesized sequences into the test tube. And then, only then, they can come up with the full genome. And this second phase, they call deep metagenomic sequencing. And once this technique is established, others are using it, and then they are coming up and say, look, we do a deep metagenomic sequencing and we'll find the sequences. And this is never proven to be an authentic result because the control experiments were lacking. It never came to the idea, look, let's take human RNA, human nucleic acid, as a control experiment, and not this one, what we consider to be infected. And this is never being carried out. And this is easy like this. And we have another sensational results. The study which was presented in a preliminary form on the 10th of January last year, finally published in Nature on the 3rd of February last year by the group around Professor Cheng in Shanghai, we can definitely show that the data they publish as original raw data from their sequencing experiments, they are fabricated. They are manipulated in a very obvious way. When you just read it, you'll find the contradiction. They say, oh, we have 300,000 overlapping sequences called contigs. And then they do their deep metagenomic sequencing. And then you should have the same number of overlapping sequences or even more, but none. They have 10 times less. And they filtered everything out so that with one software tool, you'll always get the sequence which is called SARS-CoV-2. But everybody can see who is looking at the numbers, they fabricated those data. And the second point in this scandal, in this scientific scandal, is that virologists are using 50 different kinds of software tools all over the world in order to look for overlapping sequences. This is called an assembler. They assemble sequences. But with this raw data, only with one program, a program programmed in China, MegaHit, it's possible to come up with SARS-CoV-2, and with the same data and 49 different kinds of software tools virology are international using, it's not able to create the genome of SARS-CoV-2. And there you see it's completely fabricated, and they do a lot of calculation in computers, and they claim it to us, to the public, this is the viral genome, and it's completely fabricated from the very beginning to the end. And our control experiments in the second phase were disproving the whole virology, because all virologists, when they claim a virus, and a virus is defined as a genetic sequence, they were fooling themselves, because they never carried out the control experiments, which definitely are showing that you'll get the same result with healthy human tissue.
Thank you so much, Stefan, for such a detailed explanation. This was amazing. I wanted to ask you about what you also mentioned in the Freedom Talk, that you are going to, with these results that you will have, you are going to write these letters to the government, and that you will have them translated in English too.
Yeah, I mean, now as the control experiments are ready, I'm so strong to go up with this notion in public, because it can't possibly be refuted. And therefore, I'm just on my way to write to our Minister of Health that he failed when Corona started to check the claims of the virologists if they are really scientific. And that means if the rules of science have been applied, if there's a logic, a scientific logic in their claims. And this, everybody who is confronted with a scientific statement has to show if there's a really science behind or not, or if it's just a claim, or in this case, a self-fooling of those people involved, right? Because they are not scientific. And not even mentioning those control experiments, they have proven themselves to be anti-scientific. And that's the end of virology. They themselves have proven themselves anti-scientific. And now the consequence of all of this, all corona means which are regulated by law are not legal, because the law of infectious diseases, which allows the state to put us under pressure, to wear masks, not to go out, directly be forced to get vaccinated. This law is completely useless, because it asks all people involved in this field of politics, infectious disease, to work on the actual basis of science. And the actual basis of science is that you have to publish control experiments. So as there are no control experiments in the scientific literature, they themselves prove they are anti-scientific. Therefore, the law of infectious diseases in every country is out of power. It's out of a legal possibility because the way basis is disproven. It's not only not given, it's disproven because it's proven they are anti-scientific. So all those means the government's putting up on the population are not legal because there is no science, because there's anti-scientific activities. And therefore, all those means are illegal. And this is the point, and that's the end of Corona, of the balloon, which is going to implode with this notion. I'll state this in public, everybody can use it, and that means that for everybody who can follow this argument and who check the evidence, the law is not able to oppose upon those people knowing it because virology failed to be scientific. And that's the end of not only Corona, that's the beginning, to put it in a positive manner, of developmental jump of humankind for much deeper understanding of biology, of life, of what we call diseases and how to prevent them, how to treat ourselves and people in need in a much better, deeper way. And most of all, what I can say, and to conclude here, the old good and bad thinking, which the pure materialistic point of view in science is forcing us to believe that when something is not normal or painful or deadly, that this is bad. This is over because we have a huge body of a much better understanding of life. And when you watch it, observe it, then there is no notion of a bad or a good. Life always is a symbiosis. And those processes which are turning us ill, beside of intoxication, in the most cases, there are what we call psychosomatic. And as I don't like the word psycho, I suggest soul somatic, because this is what this Platon was speaking about, or is speaking about, referring to his teacher Socrates, Plato, and that when you get ill, your soul has to be treated. And that was the wisdom which 2,500 years ago became overlaid by the new materialistic thinking. And this wisdom, which is there, which was rediscovered by a German medical doctor, Dr. Ulrike Gerd-Hamer, this is the rediscovery of what has been known before, and you'll find in the ancient Vedic texts and elsewhere, and you'll find it even in every language, that a word can kill you, a word can heal you, and so on and so on. And it's there, and Corona, it's the turning point, and therefore I am one of the few who is happy about Corona. Even though a lot of people completely unnecessarily are suffering, it's a kind of third world war. You can see it, but it's a relatively soft war, because most of the people were not forced to go in the hospital to get vaccinated. They do it voluntarily, because in this old belief. And as the majority in most of the countries are vaccinated, this is going to be the majority in future, which are going definitely to regulate and to observe what's going on in science when they learn that they were injecting toxic nanoparticles, and the RNA, what they are injecting, have no means whatsoever. The whole idea, it's crazy that a part of a virus is going to be expressed. The whole idea of molecular genetics is disproven, and to end this, I can say what I'm convinced of, that our culture is investing since 50 years, billions into gene technology, in biotechnology. That means gene technology. And never ever it is possible that something comes out which would work. It's impossible because biology, it's not working this way. It works in a completely different way. It works in a spiritual manner that a word forms the flesh, as written in the Bible. I mean, this we have, and a word can hit us and a word can heal us. And so this is one of the reasons that every 10 years or whatsoever, a new pandemic is coming up, because this is the opportunity for all those people involved to get their money out. And this is what's happening. They invested billions, never ever something is going to be taken out of. There's no return because the idea is falsified. And therefore, Corona is used or is misused to get all the money out, and in such a ridiculous way that even not knowing about virology, you just observe what they are doing with what kind of explanation, and you will see a huge body of pure contradictions and no logic. This is, what's going on.
Thank you so much, Stefan. This is really amazing. And I was thinking about it myself. And I just wanted to ask you, what is your prediction, if we can call it this way, that this whole virology will be forever disappeared, like from humanity? When do you think it can happen?
I think my feeling, I can only say my feeling and the prediction I get out of my feeling, that it is going to implode in winter. Because another thing is what is going on, because they had to define COVID as a completely new disease, because the Chinese government showed it's not SARS, it's no sudden onset of respiratory syndrome, SARS, because they showed all of their 49 cases in the 20 million city of Wuhan, 49 cases from 1st of December 2019 till 20th January 2020, 49 cases did not transmit their disease to anybody because they locked down all friends, all family members, all colleagues at workspace, all people they had contact in the hospitals, and observed them. Nearly 1,000 people had contact with these 49, and none of them got ill. So the Chinese government, proven with this, look, if it's a virus, then it's only a very difficult to transmit animal-human virus, because we closed the wild animal market, disinfect everything, and no new cases. That was their argumentation. And then comes the German professor, Christian Drosten, who constructed mentally some sequences even before the Chinese government, the Chinese scientists were publishing results on a virus or a bacterium, could be a fungi. There was not sure what they would publish and what kind of virus. But Professor Drosten constructed 3 different kinds of PCR tests. And when this preliminary data from the group in Shanghai around Professor Chang, when this was published, Drosten was sending one of the 3 sets of PCR biochemicals called primer around the whole world. And then this induced positivity in people who have not been in Wuhan. And therefore, a medical doctor came from South China to Wuhan on 20th of January and claiming, look, the Chinese government is lying because I have 2 patients, they have not been in Wuhan, but they are test positive with this virus. But this was a sequence which was prefabricated by Drosten before a sequence was published and said, look, this is our cause of disease. You must imagine this mechanism. And if you see, it's all around big business, because the friend of Drosten, who is producing the primers, they made the business because it's the company Roche who owns the patent for this quick PCR. And so they are producing the primers. This is how they did it with SARS, with Zika, with influenza, with, we have everything. We had fish and frog influenza. I don't know what. Yeah, swine flu was the last one. So, and they always are producing the primers. And this is how it happened. But in this case, Drosten was quicker than his shadow, proving that they are cheating. And so this is all going to surface because COVID is an open definition. Every person being tested and developing symptoms, everything is now named into COVID. That means the balloon is going to get bigger and bigger every moment. What means that we have a huge shift of money and power inside medicine. And so some non-educated medical doctors, they open a COVID center, have only one day the week open, but getting 10 times more money, and the money is lacking at the offices and the hospitals from specialized medical doctors. And they are the one fighting now against. So in Germany, you should see that the organization of medical doctors, they speak out. Don't vaccinate the children. Please stop every mean. But they are not heard because now we have the federal elections and everybody want to play the strong man or the strong lady, and so they are in their own trap. But this is going to get quicker and quicker. And I'm sure they can elect what they want this coming Sunday in Germany. This new government is going to step back, definitely, because Corona, it's either exploding by its own mechanism or by our force, our dedication, our brains, our hearts, our souls, our thinking. You are doing, Kate, what you are doing to spread the word, what Dean is doing. And this is what I'm convinced. The balloon, it's not exploding, which could be a little bit risk, boom, all of a sudden, but we do let the balloon implode with the argument of anti-scientific manner, anti-scientific behavior of virology. And this argument is the needle which lets the balloon implode. So that's the picture, and this is what I'm convinced. And my feeling is it's going to happen in winter. So we're going to have a really nice and springtime with full of perspectives and a humankind which is going to be much happier than before. All this unnecessary suffering, it's going to be ended for a better and a deeper understanding. And the challenge, what we have, I can put in easy words, probably in 1 or 2 sentences. If we believe in cancer as a terroristic reunion of molecules inside our body, which is able to walk inside our body and spreading the terror from one place to the other in order to kill us, to kill a human being. This idea is called metastasis. And who is believing this? Automatically it's believing in viruses, which you can spit out. It's logical. And this is the challenge what we have. And the majority of people letting themselves being vaccinated, they are the proof that they are believing into this concept. And we'll see in each society, in each country, that we have a strong number of 20 to 40% of the population who is refusing those things. And these are a lot of people altogether, knowing that the old idea of biology, which it is war inside our bodies, that our molecules turning against ourselves, that this is wrong and that we have a much deeper and a better understanding. And those 20 to 30% in all those countries, together with those who are going to awake when they realize that the vaccines never, ever could help and have a meaningful sense, but only the risk that the nanoparticles, which are highly toxic, when they are released quickly, that you are at risk to die on thrombosis or nerve cells, nerve tissue destruction. These people are going to wake up and automatically are the basis that this is not going to happen again in future, and that we finally learn. And another prerequisite, not only that we need to have a global dogma in order to change something, we have the internet. And this is another prerequisite that we can have a really global change. And this is going to happen, and this is already going. And the success of your channel, ever increasing numbers of people watching this, the quality of questions you are getting, that shows that the people are in a way to understand the things deeper, and they are going to act accordingly. So thank you very much for your work.
Thank you so much, Stefan. And of course, it, as I always say, would not be possible without you. And now with these control experiments and all these results, this means it will be the end for everything. For the COVID means, but measles means, especially that are in Germany for the mandatory vaccination, and even all the other possible like means that are taken with other so-called viruses will be over so automatically.
Yeah, this is going to be the point where the whole history is changing. And this has been predicted by the Croatian philosopher, thinker, Ivan Illich. He wrote in 1976 his book, The Nemesis of Medicine, and he showed that when medicine is part of economy and have to generate every year an increase of the interest, every year 5% plus, this is not possible with our biology, and so therefore they are forced to increase their definitions of disease, more and more treatment options. And with antibiotics, he predicted they are doing harm, not only to humankind, but also animals, poisoning plants in agriculture and the environment. And this was predicted by him in the year 76.
Thank you, Stefan. So now we discuss the end, the final refutal of virology, and to help people put all the puzzles together, I wanted to ask you about another highly requested topic, which is the tobacco mosaic virus. And, because the development of virology officially started when the Russian scientist named Dmitry Ivanovsky published his first paper on tobacco mosaic virus, which was in 1892. Could you please tell us what is it all about? What Dmitry Ivanovsky was actually doing, and what did he actually found, if it is not a virus?
Well, what is fact that every plant multiplied in a great number, they are weaker than natural plants. They are inbred plants to get bigger, quicker, which means they get weaker. And they are not adapted to every kind of soils, of course, right? They had no time to adapt themselves. They are inbred to grow quick, to raise the outcome. And that all those plants have different kind of symptoms. When they get weaker, for example, insects can easily grow on them, fungi, so therefore you have always intoxify them that they still stay stable. And tobacco mosaic disease, as the name says, they have mosaic signs on their leaves. And the tobacco, it's not that worth, because a lot of this you cannot use, you cannot smoke when the leaf is damaged on a lot of spots. And as our pure materialistic system in science explaining life and disease, it's forcing us to believe that we have a materialistic carrier of disease. And from the old thinking, it was thought to be a toxin of disease called virus in Latin. And they thought, probably we're not knowing how it looks like, but when we can filter out of diseased plants something and put it on other leaves and get the same disease, then we can say, oh, we have a filterable cause of disease, a filterable virus. And what they did, they used huge amount of diseased leaves, cut out the areas which were damaged, where there was no photosynthesis, the mosaic, and tried to get molecules out, not knowing what they are looking for. But what they are not did, they never tried to get out of healthy plants using the same amount and trying with the same techniques of filtration, what they were using then, to get in the same manner they did also an extract. So they never did control experiments. And what they did with what they filtered, they scratched it on healthy leaves. And of course, if you mechanically, with highly concentrated substances, you are damaging the leaf and you'll get a result. And even this was not controlled. So that they used other substances, what they filtered from completely different kind of plants, and harming those leaves in the same manner. So that would be the control experiment. And this had not been carried out. So there was also this self-fooling. There must be a virus when there is a damage, because there are no other explanations possible in this system. It comes not to their mind, oh, the plant is not adapted to the soil. Where are the diseased plants? When we grow them elsewhere, are they better or what? So no systematic science on this, only the focus, there must be a materialistic carrier of a disease, of tabac mosaic disease. And so they called it tabac mosaic virus. And in this system, we also have the evolution of the idea. Before it was a toxic protein. And later, after 52, they said, oh, it's the nucleic acid, which is the virus in what they isolated. Before they ignored the nucleic acid. And they said, no, it's the virus. And after what they found, all or virtually all nucleic acid in nature, especially the DNA, our chromosomes, are wrapped around proteins. For example, in chromosomes, they called histones. It's known, you know. And this mixture from proteins and nucleic acid, this was considered to be the tabac mosaic virus. Yeah. And if you'll check the evidence, no control experiments carried out, documented, and you'll see there's no science behind this. And it's obvious what is making the plant weak. And this is the result of agriculture to multiply plants in a high number to get them all uniform, to get the maximum output, and the price what you are paying, that they are not adapted to the soil when minerals are lacking, when the soil is too, it's not balanced for these things you need in all soils. You need a huge amount of microbial life, you need a huge amount of fungi, you need bacteria, you need insects, you know, working in the humus. This is a living sphere on its own, this 30 centimeters around our globe on the earth, the humus. This is the living skin of our planet, and all factors inside there are needed to create a healthy plant. And if we misuse, and even not knowing what is going on in the earth, in the humus, then it's no wonder that we are creating weak plants which easily are going to die, or which are not growing in the way the people want to have it. That's the story of tobacco mosaic virus. If you see it, and also when later on they come up with the sequence of this virus, you'll see the same self-fooling in the construction of the genetic sequence of tobacco mosaic virus, the same manner like it was done now with SARS-CoV-2.
Thank you, Stefan. It was extremely clear. Thank you. And I also had another question, which is rabies, which is another important topic in this context. Rabies was allegedly isolated by Louis Pasteur in 1884. And rabies is actually very important topic because it is the same as COVID or HIV, but for animals. It is their COVID, so to speak.
Well, look, I mean, COVID, it's an open definition. It's an open symptom complex. It's ever growing, whereas AIDS or rabies are only a few symptoms, right? Which are said to be AIDS or rabies when you test positive on HIV or on the rabies virus, right? And what Montagnier did at this time, in the old virology, a virus was considered to be a toxic protein. And it was said that the virus can grow only in the same tissue which the virus is damaging, so nerve tissue. And what they did, animals which cramped, which we nowadays call epilepsy, right? And which is in the healing phase, a healing crisis, which with all kinds of disease are happening. And if the motoric system is involved, an accident, an act of violence, war, everything what can happen and what people are doing against them, also with animals. If they heal out, they cramp. This would be a short one, or, you know, and especially in children of or soldiers in war, you see, you know, where the explosion came from when they try to protect them. And when this comes up, or they stay. So this in animals is all called rabies, or when they cramp, when salvia is flowing out. We have a lot of different somatic explanation, exact explanations why humans or animals react in this way or another way. And this was considered to be a virus. So out of the brain of diseased animals, they isolated something, and Pasteur did not even isolate. He extracted huge amount of brain and shot 30% of the volume of a brain into the brain of a dog. And of course, it was a German shepherd. Pasteur was hating the Germans, and so his hate was focused on the German shepherds. And the dogs were erected onto a cross, and they could not move. And then you extract 30% of the volume of the brain through the ear into the brain. Man, sure that the animal is cramping and you know, all kind of disease. And this is the quality of the experiments Pasteur and others were publishing. You have just to read it, and you see it's pure, it's madness, and no control experiments carried out in a way that you just inject 30% of pure water or pure ringer solution into the brain, and the same thing would happen. No control experiment whatsoever. And there's a good review. It's in German language, and it's published in the year 1999 at the Max Planck Institute for the History of Science. And it's a review with the German title zur Frühgeschichte der Virologie, on the early history of virology. And there you'll find everything cited that virology, the old virology, gave up themselves because they carried out control experiment. They did exactly the same, but with completely neutral liquids, injecting into the brains or into the lungs of animals and so on. And they got the same symptoms. And therefore, you'll find in this review on the old virology that till the year 52, no single experiments of infectivity worked. To the contrary, it was never ever able to induce the disease, even on tobacco mosaic virus. When you did a normal experiment, you would consider, oh, this is it, to get the dust of infected material on the plants and not to harm them mechanically, right? So you'll find in this review that all viruses, and they had all been defined, polio, measles, blah, 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 with none of them transmission of disease had been achieved, had been possible, to the contrary. So only after 52, the idea of what is a virus changed, but the fact that a transmission of disease with polio, measles, influenza, whatsoever, never ever had been possible. But they are not speaking about this. And that means the scientists are not only anti-scientific, they are not honest. They are not speaking out what is there in the scientific literature of a highly respected scientific institution, the Max Planck Institute of the History of Science. It's there, but they are not speaking out about this. Yeah? Even it is a law of scientific work, a good scientific practice, to cite everything which is violating your own belief systems. You have to cite the one who are criticizing you. And this is what they are not doing. This is just another proof of the virologists, and not only virologists, being highly anti-scientific. They are not following the other laws of science to cite findings, experiments, which are contradicting your own belief system. And this is another law of good scientific practice, as it's published in 1994. And they are not just citing this, which is there in the body of the scientific language. This is a review of 98 pages, and it's in German language, and with all those technical means to translate those articles in 1 second in every completely different kind of language. This is not justifiable to ignore such a huge body of information, but of course, it's difficult to question yourself. To end this, it means to be a scientist means to be a little bit masochistic. You have to try to disprove yourself every day. And this would be real science. And this is what they are not doing. And it's understandable that they are not doing this. And this was already published in the year 1929 by Eugen Rosenstock, who is one of the greatest thinkers I'm aware of. He said that they are doing pseudoscience, and he really said he is criticizing cancer and infectious diseases. He says, and it fits perfectly with rabies, with your question, he wrote down in 29 that they are doing cancer research on the old ideas, means falsificated ideas, of Pasteur, as would it be rabies. He definitely showed that all the thinking of cancer, that some bad molecules are there and then they're killing yourself, ever growing, that this concept is false, and it's based on the idea of infectious diseases. So thank you very much for this important question on rabies, and probably you should translate those texts from Eugen Rosenstock you see into Russian and to all other kind of languages, because it's of greatest importance. And I can tell you, this thinker is just such a great human spirit. He made me very optimistic because his view on history, it's so great, and it's worthwhile to study it and to incorporate in your brain, in your heart, in your soul. Is just great. And I guarantee you, every page of his books you are reading, you are clapping 3 times into your hand, saying, thanks God, coming across this deep understanding of life and our history. Eugen Rosenstock, Hussey, on Pasteur, cancer, and rabies, for example. In his work, Sociology #1, in the year 2008, it was finally published in a way in 3 books like he wanted. In 56, it was published in 2 books, but now we have an edition in a way he wanted this very important books to be published. It was published in 2008 at Thalhammer. Thalhammer. You'll find it on the internet.
Okay, thank you so much, Stefan. Is it available in English?
Yes.
Okay, so I will make sure I'll find them. Yeah, thank you so much for explaining this about rabies. What I meant when I said they are like COVID, of course, diagnostically not, but with means that are applied to animals, all these vaccine passports and microchips that you need to prove that you have this vaccine, the rabies vaccine for your animal. And then it's not enough. You also have to test them. The same as it is with COVID vaccine. It's not enough to have the vaccine. You have to test. In some countries, you have to do the quarantine, and you need what they want to do now, this passport and all of this. This is what I meant. Do you still have a little bit of time?
Sure, yes.
Yes, I just wanted to quickly ask you about one very, very requested thing, that is graphene in the vaccines. I know that it is like a secondary, I always say that, because I think people invest too much time into thinking about vaccines and the contents, because the main topic must be the virus and what we're talking about right now. But still, because at least once a day I get this question, do you have something to say about this?
Yeah, it's a challenge, but I don't like this discussion because it distracts from the real dangers with these corona vaccines, the nanoparticles. So nobody speaks about nanoparticles because there's a hype that gene technology works, and that's the RNA, and then other things. I mean, this is so crazy. In the year 2009, 93% of the German population refused this vaccine. So they had to burn it in millions. In Magdeburg, in the German city of Magdeburg, they had to burn the swine flu vaccine, and everybody survived. They just stopped to talk about. But what happened? They published the issue of the Bundesgesundheitsblatt, the journal of the Federal Ministry of Health. They issued 12, 2010, Pandemia, Lessons Learned. Why we failed. But they were not thinking, hey, the virus was there. Nobody got vaccinated. Nothing happened. They just stopped to test, stopped to talk about, and that was the end of swine flu. Without any vaccination, 7% of the population, 7. Look, and but then they said, look, in the future, we should take care. We should impose censorship on the internet. This was discussed and asked the internet companies to impose a censorship. Even though it's not justified in the laws of infectious diseases that we're now carrying out censorship. It's illegal. It's anti-constitutional what they are doing. But our government, the federal government in Germany, asked with this journal the companies to carry out censorship. So that no wrong information, it's withholding the people to get vaccinated. And this graphene discussion is distracting from the fact that we have the same very dangerous nanoparticles inside the vaccine like we had in 2009. There we had only one form of nanoparticles. Now we have 4 different kinds of fatty acids in the form of nanoparticles, and only one of the 4, polyethylene glycol, PEG, it's known and started, and it has a highly allergenic potential. So it means when you get a shot and you think, oh, this is going to kill you. Or even after some days when you think, oh, you can move your arm or your whole body, or your brain is exploding. And in this moment, when you got into a shock and the shock is lasting, then in the very moment, the body recognizes everything. Smell, color, sound, surrounding, even persons, what you have in your mouth, and of course, what you have in your muscle. And then you react allergic in future when always you get in contact with those substances being around you or inside when a shock program starts. And that's, then you have an allergy. And what they are not knowing, the people, this PEG, it's in all those cheap soaps and douche shower gels, all those highly toxic what we are putting on our bodies, in our bodies, and in our environment, there it's inside. And then the people not knowing, oh, what's going on? And it's then you test positive, it's called COVID. Skin rash, whatsoever, allergic reactions, you know. And graphene is completely points to a wrong direction, because graphene, it's used in cancer therapy because it's not to produce antibodies. That means a humoral reaction, what they want to have with their vaccines. They want to increase the production of globulins, small proteins, which they consider to be antibodies. So, and graphene, it's not doing this. Graphene has a cellular immunity. That means it destroys the tissue as a whole. This is highly toxic, and it's used in cancer. And they are doing put it onto iron, so that they inject graphene with iron into your bloodstream and then get a strong magnet on over the organ where they want to hold and concentrate the iron in order to destroy the cancer at this certain place in your body. Therefore, graphene is used, but not in high concentration in those vaccines. What they are using, nanoparticles out of carbon, like graphene, it's to smoothen the rubber gum in the syringes. Because those syringes where the vaccine is inside, they are stored with, sometimes some at -50, some at -20, and they stored for months or years whatsoever. But when you use the syringe, it must always work smooth, that you are not injecting it in 1 minute, because this would destroy mechanically too much, and the vaccine would run out of the body, or destroy mechanically too much when you inject it too quickly. Therefore, the rubber stamp inside the syringe always have to go smooth, and therefore they're using graphene and similar substances to get it lubricated. So even after storage for long months or years, even frozen, that this works. And so you find traces of them in all medical products where they use rubber. I mean, but then to claim, look, it's there and it's illegal. I mean, how stupid it would be, because a lot of people are stealing vaccines, a lot of medical doctors not injecting them really. They pretend to do or use an empty one. Or I was working in a medical office, and then I saw, hey, the hell, at the sink, the sink was eroding, and I was, what kind of aggressive substances was used there. I looked in the medical practice, other things, no. And this was the room where the medical doctor was vaccinating the people. And once he showed me how he's doing it. So in case he's not there, occupied, that I should vaccinate. And of course, I never did, you know. And then he was just throwing it away. He stand behind the patient in order to vaccinate him and threw the vaccine substance inside the sink. And sometimes he was too far away, so the liquid were running above the sink and destroying it because it's highly toxic what is inside. And this is what I observed. So a lot of vaccines are around, you know, and are sent into the labs and to analyze what is inside or not. And those claims that there is a highly amount of graphene in order to intoxify or do some experiments in this thing. I mean, this is so stupid and so stupid. None of the pharmaceutical producers are going to be. It's impossible because the vaccines are there and they are tested for this, and those high amounts of graphene, which are claimed are inside, in order that they are going to be toxic, they are not inside. That's not true. And all claims which are inducing fear and which you cannot check if it's true or not, they have the function to block you and to distract from something else. We have to have fear, but for given reasons. Big fire, don't get close to the volcano on the island La Palma, you know, especially when it goes into the water. So those fears are there and are useful, but fears which are not real are very, very dangerous because they can cause psychosomatic, I mean, soul somatic processes, which can be dangerous in the long run, you know. And therefore, I ask everybody when he gets an information which is inducing fear, but you'll not get in the same time the tool into your hands, into your brain, into your heart to deal with the fear, then you really should consider to think twice if this is real or not. And a lot of people who have the will to help, to avoid people getting vaccinated, they are increasing the fear in order to help the people. It's a mechanism. So, but this fear is always rising and itself very dangerous. It's always very tricky to use arguments which are inducing fear. And graphene is one of the examples, which itself it's very dangerous and it's distracting from the real dangers of those corona vaccines, the nanoparticles.
Thank you so much, Stefan. I could not say it better. I fully agree with you, and this is what I also felt myself. That's why I did not want to engage in these discussions. And I want to thank you for your time. I have all my questions finished. I'm extremely grateful for you and for everything you do. And thank you for your wisdom. And I just, I'm amazed. Thank you so much.
Yeah, thank you. And thank you, Dean. You're doing a very important work. Without you, my findings or my work would not have any effect. I mean, we belong to each other. We form a group and we'll do what everybody can expect from us as human beings, as citizens, and as responsible human beings. And I think this is normal, but I mean, it's so great how you are doing it. Also, your mother doing nice graphic. This is also very important how it looks on the internet. So thank you all. Thank you, Dean. Greetings to your mother and to everybody and to all the people seeing this. Please check the evidence, incorporate it into your brain, into your heart, And then if you feel good with the information, then spread it and act accordingly. Thank you very much.
Thank you, Stefan, and thank you, Dean. Thank you so much.